social implications beyond designer babies. It can be used for somatic cell gene therapy. It can also be used to eradicate vector-borne diseases, and that's one of my main interests in Texas. That's the use of gene drive, and has some biosecurity implications that I'll talk about later, especially for food security. CRISPR array and its associated loci constitute a unique prokaryotic immune system that is poorly understood. Its origin and evolution present problems for the neo-Darwinian interpretation of evolution, and that story will occupy half of my talk. In what is called CRISPR gene editing, a CRISPR-associated protein, most often it's a protein called Cas9, is used with a guide RNA to edit genes with very high accuracy. This technology enables somatic cell gene and germline changes. It also enables gene drives against vector-borne diseases and food security attacks. This story will be told to the extent that time permits. Where did CRISPR come from? In 1989, Yoshima Ishino notices a CRISPR sequence in Escherichia coli in a laboratory in Osaka. Francisco Mojica in 1993 reports CRISPR sequence in Archaea. He's working in Spain at the time. In 2002, the term CRISPR was introduced 10 years ago by Janssen et al. And in 2005, 3 groups independently recognized the similarity between the CRISPR spacer sequences and those of bacteriophages, archaea, virus, plasmids, and this automatically suggested them that the function of CRISPR was to confer immunity against these systems. That hypothesis was already around by 2006. And then 2007, there was an experimental demonstration that CRISPR-Cas systems provide immune resistance to viruses and bacteria. And by 2012, Jennifer Doudna and Emmanuel Charpentier had used CRISPR-Cas9 for protein for targeted gene editing. And that's the source of all the biotechnology uses of CRISPR that we're going to talk about today. What are the CRISPR arrays? This consists of a leader sequence followed by 2 to 100 repeats, each 25 to 35 DNA base pairs long, interspaced with spacers, which have highly variable base pairs each. The repeats are partly palindromic by base pair complementarity. There's palindromic not if you associate a T with an T, but if you associate a T with a C, a G with a C, a T with an A G with a C, an A with a T, and so on. So you are using base pair complementarity to suggest that there's a palindrome over there. The spacer sequences, those are the ones in between the repeats, are highly variable, and they're derived from invaders, such as viruses and plasmids. They actually constitute an immune memory. They keep track of the fact that an invasion by these invaders have happened. When a new invasion happens by the same virus or plasmid, RNA derived from the spacers are used to recognize the invader's protospacer. That's the original DNA sequence that has been inserted into the variable regions of the CRISPR array. Accompanying Cas proteins then inactivate the invader. In particular, Cas9 very effectively cleaves RNA. This entire process is usually said to have 3 stages. One is called adaptation, the second is called expression or processing, and the third is called interference. In adaptation, a complex of Cas proteins binds to target DNA in the invader. This requires some self-non-self discrimination. We don't completely understand how this is carried out, but it is. The complex migrates down the molecule until it encounters 2 to 4 base pair motif known as a protospacer adjacent motif. It cleaves out everything next to the PAM, that portion of the target DNA, and then it inserts it in the CRISPR array between 2 repeats, typically at the beginning of the array. Just that way the array gets extended and new memory is introduced because this new spacer sequence has been introduced in the CRISPR RNA. This is the most common mechanism. Some CRISPR systems acquire their spacers not by cleaving DNA, but by reverse transcription of RNA. When we turn to expression processing, the CRISPR array is first transcribed into a long single transcript, the pre-CRISPR RNA or pre-cRNA. This is processed into mature cRNAs, each consisting of 1 spacer and part of an adjacent repeat, and one of these typically remains bound to the processing complex, and that with the Cas proteins. In interference, the cRNA is bound to the processing complex is used as a guide to find a region of the DNA next to a PAM. The PAM is then required for proteins such as Cas9 to latch on and cleave the DNA. The invading DNA is thus cleaved and deactivated descendant cells will continue to have this capacity of recognizing this DNA and deactivating them. It's similar to the PV system in eukaryotes, very, very similar, in fact, and that might be telling us that there's some basic principle of immunity that we are seeing here expressed in this form. And that's a picture over here. We have the Cas9 protein. You see the adaptation stage, and then finally you see the interference in the stage at the end. The power of gene editing using CRISPR arises from its accuracy. It's unprecedented compared to previous technologies. We often call them BC technologies, where BC stands for before CRISPR technologies. It's very low cost compared to other technologies. You can buy a CRISPR system for about $200 on the internet and do the experiments in your garage if you want, and people have done that. And it has a unique flexibility. You can target any gene whatsoever using the same system. All you have to do is create the appropriate CRISPR complex, which is very easy to do. We turn now to the biology of CRISPR, why it's interesting. One is that the evolution. It's interesting. Does it challenge the neo-Darwinian interpretation of evolution? And then the immunity, the unsolved problem of how non-self discrimination is used when the bacteria first decide that they will use CRISPR to recognize and incorporate foreign DNA during the adaptation process for all of this, right? I won't talk about the latter process today. I will focus on the first process. Does it present a challenge to the neo-Darwinian interpretation of evolution? Recall that the acquisition of immunity through CRISPR-based mechanisms is very, very important. It appears to be a clear case of IAAC, inheritance of acquired adaptive characters, which clearly challenges the received view of evolution. It's not just that it is inherited of acquired characters. That, to me, is not enough to suggest that there's something really wrong with the neo-Darwinian picture. It's acquired adaptive characters. The process consists of adaptation, expression, and processing. This takes time. If you think all of this is happening in a bacterial cell, won't the bacterial cell have been killed by the virus before all this can happen? That it recognizes, it adapts to the virus. There's expression processing of the single CRISPR transcript so that you actually have a functional system, and then the functional system carries out an interference. And we do not know the answer to this, but what we know from the studies of how much time the process takes is, yes, if that's what you think is going to happen, in the single cell is going to adapt in a heritable way to invasion by the CRISPR by incorporating CRISPR into its genome, there simply isn't enough time for it. A cell cannot do it that fast. But it's not just the cell that matters, it's the lineage. And that's what I'm going to talk about now. And this is based partly on solid empirical results. They're fully testable, but not every aspect of what I am about to say, which is the most likely scenario of how CRISPR works, according to most experts, has been completely proven. So what I'm presenting now is a hypothetical model. The empirical facts we have is the following, that when, for example, a population of viruses infects a bacterial cell lineages, they have some defectives. There are some defective viruses present in the populations, and these cannot replicate successfully. And the other piece of evidence that we have is that the fraction of bacteria that acquire CRISPR-based immunity closely agrees with the fraction of defective viruses that are known in those kind of viral populations. This suggests that acquisition of CRISPR-based immunity somehow relies on the presence of defective viruses. So what do you think happens? When viruses invade a bacterial population, most of the bacteria or the archaea, they just simply get killed. Sometimes a lucky cell gets invaded by a defective virus that cannot kill it. It then begins the process of adaptation. After completion, if the same virus invades again, it can launch into expression processing, but this is not very likely. Typically, only cells that will be in this environmental context are its descendants. The same cell that was lucky enough to get a defective virus is unlikely to be invaded by that virus again. Its descendants, though, are likely to be invaded by viruses, and that's the stage at which we have CRISPR-based inherited immunity, right? But is this adaptation? If the bacteria encounters the same virus again, yes, this is adaptation, but it'll problematize what same means later in the talk. We're going to talk about that in a few minutes. If not, we must face up to the fact that to the problem of the unit of adaptation. If the unit is the individual cell, no. There is no challenge to the neo-Darwinian interpretation of evolution. If the unit is the lineage, yes, the lineage is the thing that's adapted, and perhaps lineages are the proper units of evolution, and I'll leave that for the discussion. In fact, since I have time, I will say two things in favor of the idea that the lineage is the unit of evolution. One of which is that when we begin to say that populations evolve, there is a metaphysical problem there. Think of annual plants or something like that. The actual population that you see at any given time where you have variation and natural selection is taking place just dies. It's that the next population, which is metaphysically different from that population, that you begin to see signs of adaptation. So I think actual talk about populations evolving is not really accurate talk. What really is evolving are the descendant things, the diachronic things that are going across from one generation to the other, and that's a lineage. To this, I'm going to add a technical result that has come out from several groups over the last few years, not my own group, which is that if you take the lineage as the unit of evolution and you just compute its fitness, right, its natural fitness of the lineage, that turns out to be the inclusive fitness of the cells in the lineage. In fact, if you take the lineage as the unit of evolution, natural selection and kin selection are the same thing. Okay, it's not individual selection ever, it's always kin selection and all of it. And that by itself is very good reason to think of the lineage as a unit of selection. And in fact, there's a dissertation waiting to be written just on that topic of how much more simple the theory of natural selection becomes if we move away from individuals and populations and treat lineages as the units of selection. And now along comes CRISPR, and now you suddenly see what's happening in CRISPR really looks like adaptation that is taking place at the level of lineage, which gives you one more argument to think in this way. And that is the most important reason why CRISPR, beyond all the biotech hype, is biologically interesting. It leads us naturally to the view, the lineage view of evolution, and lineages being the unit of selection. But there's still problems. What is the environment with respect to which it is an adaptation? If it is a defective virus, then it cannot be an adaptation to it because it was completely fit with respect to that virus. The virus was defective. It couldn't kill it anyway. So what's the sense of that? If it's an intact virus, it was not exposed to that environment. So it isn't as if it's being adapting to some environment it's being exposed to. So we have a paradox here, and the solution seems to be, again, generalized. The relevant environment is not the virus. It must be the population of viruses, including the defective viruses in it. One more complexity that emerges. I think because of all of this that you can teach an entire philosophy of biology course just around the CRISPR example, because every interesting thing that comes about the nature of evolution and selection all arises in the context of CRISPR. Is it Lamarckian? And there, of course, the issue is between whether it's the inheritance of acquired characters and the inheritance of acquired adaptive characters. Throughout the 20th century, Lamarckism was identified with the inheritance of acquired characters. This is clearly inappropriate on multiple levels. I can spend hours talking about why that's inappropriate, including the fact that everybody believed in the inheritance of acquired characters, except briefly Darwin, and then it really gets challenged by Weizmann. There's nothing special about Lamarck there. I have talked about this since 1991. I really think we should stop worrying about this particular question. Whether or not something is Lamarckian or neo-Lamarckian simply is not that interesting as a question. Is it an inheritance of acquired adaptive characters? Well, the problem was adaptive. I noted earlier, we shelved that. We also decided that there's no point in worrying about whether or not we should call it Lamarckian. Perhaps what really divides Lamarckian from neo-Darwinian is the reliance on determinate versus stochastic mechanisms. Add to that the thesis of the blindness of variation that refers to the separation of the processes of adaptation and the generation of variation, then yes, we are seeing something that possibly be Lamarckian, but really the issue, the proper attitude might be to simply ignore the question of whether or not something is Lamarckian altogether, because it's just not that interesting. That's all I want to say about the biology of CRISPR today. I want to move on to the ethics of CRISPR. As far as the biology of CRISPR goes, I have not talked, I can take this up later, about the self-non-self discrimination But it's not clear to me that I can say anything interesting about it because it's simply not understood. But clearly this issue of lineage selection is something that is worth thinking about. As far as I know, nobody has yet published anything about it, so it's particularly worth thinking about. All right, let's turn to gene editing. And here I want to make a huge distinction between somatic cell gene editing first starting out with non-human species. There is no ethical problem here. It's like ordinary intrusive intervention. In non-human species, safety is of paramount concern, and whether or not you use CRISPR makes no difference. You can carry this out in a variety of methods. Trouble, of course, here is when you get into germline gene editing, even in non-human species. One of the standard use of CRISPR, Cas9, is in agricultural contexts, and this has been done systematically with livestock being edited with BC methods as well as by CRISPR, particularly in China, particularly to grow pigs faster and have better pork, because those are important given the diet and the population of China. And a large class of problematic results have emerged, almost all of them, when a particular gene called the myostatin gene has been targeted for modification. In pigs, it leads to leaner meat but extra vertebrae. In rabbits, it leads to more meat but enlarged tongues. In fact, the tongues loll out to such an extent that the rabbits might even die of suffocation with their own tongues. In lamb, it leads to change wool color, but not always the predicted ones, and cesarean births. 40% of the pregnancies end up either with cesarean births or in death of the mother. So from an animal welfare perspective, CRISPR-based germline editing has been a complete disaster. But in societies that do not have scruples very much about animal ethics, that is not a problem. In societies that, say, the European Union, where people are very, very concerned about animal welfare, this germline gene editing is even a problem in non-human species. When you get to human somatic cell gene editing, again, there really is no ethical problem here. It's like very intrusive medical interventions. It's like brain surgery or something, heart replacement or something like that. It's not going to go off into the next generation because it's only the somatic cells that are being targeted. CRISPR actually makes it likely that that finally somatic cell gene therapy is going to be successful. And recently, there already has been some success in the case of sickle cell disease and a few other diseases like there, where somatic cell gene intervention has actually been successful. If you are getting worried about that, well, yes, it's only somatic cells, well, there's going to be foreign DNA inside you. Well, every time you get an organ transplant, there's foreign DNA inside you too. I mean, there is nothing special about any of this. The ethical worries are all hype mainly. The real problem with somatic cell gene interventions is how do you deliver the genes to the intended tissues and organs and nothing else. And this depends on whether or not you can find safe viruses that target specifically those tissues and organs and nothing else, and whether those are effective. And we have quite a few that are reasonably effective. It's not impossible. When you get to human germline editing, and usually when I'm giving this talk, people have this finally expression in their face. This is where CRISPR becomes ethically interesting and very, very problematic. It's the specter of eugenics, and it would be eugenics. You are interfering in the gene pool of the human population, but then you have to really ask yourself, is eugenics strictly undesirable? And I think that's a question that we need to answer. Because whether we like it or not, the generation of anybody here in this room who's less than 30 years old is going to face this choice, that when they reproduce as to whether or not, if they are having reproductive difficulties, and they are going to rely on IVF fertilization, right, in vitro fertilization anyway, when that happens just as a certain number of these fertilized cells get discarded because they have genes that are considered to be dangerous, wouldn't you rather take these cells and actually change the DNA in them to make them not dangerous? And if you happen to be, as some people too many people in the United States happen to believe, that these fertilized cells already are human beings, using CRISPR-based gene editing, germline editing, actually is an alternative to something that they find as problematic as abortion. So there is a real issue here over there that has to be faced up. But the real thing to remember in any context of eugenics are the two things. One of them, it's very difficult to define. And the other thing is that eugenics became unacceptable because of what happened in Nazi Germany. The history of eugenics really starts in the United States with sterilization efforts that Hitler very, very much admired. And he thought was the best contribution that the United States had ever made to civilization. And then and Hitler was basically following the United States and mimicking it when it decided to go through and carry out, and introduce and emphasize the Nazi program of eugenics. What do we mean by eugenics? You're targeting a human phenotypic trait that we want to promote or discourage, sickle cell disease, Huntington's disease, something like that. We endorse conscious intervention in individual reproduction. We interfere at the level of the fertilized cell or the early zygote and change it at that level. So if you're at the stage where you still have stem cells that are truly fully pluripotent, and we take one of the stem cells, modify it, kill the others, the one that we've modified then reproduces, creates the other cells, and an abnormal embryo for which the germline and all cells have been modified gets created. The purpose of a eugenic program must be to change the future distribution of the trait in the population as a whole, not just in individuals. And what people nowadays are defending more and more is what's called liberal eugenics. This is all parental choice, but you can be a bit stricter than that and also ask for some state intervention in a program that's called moderate eugenics, where, for example, if you're providing health services to individuals that are paid for by the society, then the insurance providers or society as a whole can demand that certain genetic diseases be eliminated just for the sake of cost optimization. Okay? And there are usually 4 objections to all of this. We are playing God if you interfere with the human gene pool. I don't know what to make with this argument. We are playing God all the time when we interfere with things. And maybe we are, but what's wrong with it? Just because we are playing God does not mean we are doing something wrong, right? Human heritage, that we are destroying the heritage of humanity. Well, look, this relies on a very bad analogy. And you might just say when we are ending slavery, we are also destroying the heritage of humanity. Once again, this is not a good argument. There's no informed consent. The fertilized cell has not agreed to have its genome changed. But we do that all the time when we surgically intervene with fetuses, children, and others where parents make decisions for the young. I mean, of course there's no informed consent, but we accept no informed consent at all in all kinds of other situations. The really serious one is disability rights. Society decides that a gene has to be eliminated, and the people having the gene might think that they're being targeted with no reason whatsoever. What do I have in mind here? There is a condition or a whole family of conditions called hereditary deafness. And most people who are not deaf think of this as a disability, and it has to be eliminated. On the other hand, most people with hereditary deafness today think of just being different from other human beings and not having a disability, and a large fraction of them prefer to have children with hereditary deafness rather than without. Hearing children, and the hearing children of such deaf of individuals often think of their deaf siblings, cousins, deaf community members as just being different from them, but in no sense having a disability. And that's the kind of problem you have to worry about if you say, or even if you encourage parents to get rid of a particular gene by germline intervention and remove it from the human population. And I take that objection very, very seriously. All right? And there's a huge distinction here to be also made between what the purpose of germline intervention is, whether it's genetic enhancement versus disease gene elimination. This corresponds to the old distinction between positive and negative eugenics, and the problems outweigh benefits of gene enhancement completely. First of all, it's biologically implausible. I spend a lot of time in the book explaining even with such ordinary physical traits such as skin color, hair color, and eye color, how complicated the genetics actually is, and how almost impossible it is, even in those cases, to suggest intervention at the genetic level. Most importantly, though, that these traits are usually going to be selected purely because of social prejudice. Who says that the ability to run better is better than the ability not to run that well. Who says that intelligence is that desirable if a lot of very intelligent people are usually very unhappy? And IQ, in the case of IQ, we all know that its history is a history of racism. So, and these are all things, so we don't even, the choosing things to enhance is often going to be nothing more than a reflection of reified social prejudice. And also, even in circumstances like that, we have to deal with it, that there is a problem of who's going to have access to this technology. And this is in general a problem with biotechnology that Stuart Newman and people like that have been emphasizing for a long, long time to worry about it. So we could have a complete ban on genetic enhancement. That's what the situation is right now. It's the right thing, but then you might say that it is actually interfering with reproductive freedom. That's what lots of libertarians in this field of reproductive freedom, including some colleagues of mine who are now deceased, have been arguing for ages. But there's also an issue. Now, who says that complete reproductive freedom is necessarily a good thing for society to have? All right? I mean, we deny complete reproductive freedom, for example, in the case of incest. Okay, if we can do it there, why can't we also do it somewhere else? Where we say that no, you cannot have a child of this kind. In the case of disease gene elimination, though, these arguments are not that clear-cut. If safety, accuracy, and specificity, which means it will work, and the change that will happen is the intended change and nothing else will happen, that if these are satisfied, it's hard not to accept that germline intervention may be ethically possible. Certainly, this is a subject that needs further discussion and policy formulation. My own conservative suggestion right now is that the only condition that I know of that's relatively common for which germline intervention is justifiable is myotonic dystrophy. It's a single dominant gene that causes it. And maybe Huntington's disease, but even there are problems with the complexity of the sequence of mutations that can cause Huntington's disease, but sometimes it might be possible. Let me turn to the final use of CRISPR, and this is the last topic that I'll spend any significant time on, and that is the use of CRISPR in order to spread genes across a population and thereby perhaps drive a population, if not the species, to extinction. These are called gene drives. What you do is you take a CRISPR construct and attach it to something that is like a selfish DNA. You insert it in a particular point in one chromosome. Because of the selfish DNA, it inserts itself into the other chromosome. So the next generation, all the descendants will have it, right? One copy of it. Because of the selfish DNA, it will be inserted in the other copy too. In a few generations, much faster than Mendelian inheritance, it'll spread all across the population. These are called gene drives, right, over there. And the idea that we could use gene drives became very, very attractive about 7 years ago when Zika was a major problem. This was before the pandemic, when we were afraid of it spreading, because a colleague of mine who now teaches at Texas A&M, was in Virginia Tech then, Zach Adelman, found a gene that if you used it on Aedes mosquitoes, the ones that spread dengue, Zika, and all of that, then these mosquitoes are entirely turned male, okay? And if mosquitoes become male, that leads to two consequences. One of them, they don't spread the disease themselves because they don't bite humans. Only the females bite humans when they're gravid, when they have eggs in them. And moreover, if a population entirely turn males in a sexually reproducing species, the population goes extinct. And these are it. Standard gene drives can spread all through the population, but you can also have what are called daisy chain gene drives. When one introduced gene ends up creating another gene that helps create a third gene or a fourth, and is the final gene that's effective. And you can use CRISPR constructs for all the first few all the sequence of genes. And in the case of the first gene, design the construct in such a way that natural selection will ultimately remove it, so the CRISPR reproduction naturally comes to a stop. That's the daisy chain gene drives. And they are, for reducing fitness, when they reduce fitness, they're of great relevance for disease vectors. For example, for Aedes aegypti, classical biological control has not worked. Pesticides are environmental and human health risk factors in Brazil and other countries. Inadequate water supply and poor sanitation generates open containers of water for mosquitoes to breed in. We have not been able to control it. Rio has done a fairly good job by trying to get rid of open water containers. Control has happened at the sociological level, but this often works in disease situations and all of that. But by and large, it hasn't worked very well. Why not use a gene drive? And Zach Adelman and I have been arguing this for 7 years now. I suggest that they should be used. He suggests that they shouldn't be used because it's too risky. We might end up driving a species to extinction, and that's necessarily a bad thing. There are, of course, pragmatic problems with all of these. There are many, many uncertainties in the process. But what Adelman and others are worried about are in-principle problems, deeper issues of what can and cannot done when we have the ability to do something. For example, questions of whether or not we have the right to make a species extinct by choice just because we think it's a harmful species. And people are worried of a slippery slope. I say do it with mosquitoes. The next person comes along and say do it with rodents. Where do we stop? And all of that. People have suggested using gene drives, for example, to eliminate feral rodents from island populations. So this is not, the slippery slope is out there. It's not just purely being invented by philosophers who have nothing better to do. There are practical problems. How effective are they? The long-term stability, the unique importance of species, assumptions about dynamics. There's the irreversibility if something goes wrong. There's always unintended consequences when you're worrying about specificity. The logical conclusion would be to eliminate species. And then, of course, if we eliminate one species, what we end up eliminating scores of them, their unintended consequences and all of that. We should proceed with caution. You need proper ecological risk analysis, and just looking at molecular biology is not going to solve the problem of what we're doing with gene drives. We have to make sure that the ecological risk analysis has been done, and then we have to ask ourselves whether or not the benefits in terms of human suffering outweighs all of this cost. But as I keep on pointing out with gene drives that we might be making, this might all be much ado about nothing. That is that gene drives in practice, when people have done the experiments, typically they are selected against. And one consequence of being selected against is that they never end to a complete exhaustion of the species. You end up in a situation where, because of the selection, the gene drives stop operating after a certain amount of work. So we might not really have anything to worry about in it. All that gene drives can do is reduce population levels, and even getting rid of a complete population actually requires gene drives to be supplemented by some other methods, such as pesticides and all of that. And even then, we only end up eliminating populations. The idea that we would eliminate a whole species is almost entirely fictional. The last example I typically give at this stage is that of not so much of gene drives, but of another thing you could do with gene drives, not so much with disease, but with food security. So one of the things we learned from the pandemic, particularly in places like Austin, where I'm from, is suddenly all kinds of food that we typically thought would come from New Zealand and God knows where just disappeared. And we had to worry more and more about what was locally produced. And with all these links, trade links being disrupted, we had to worry about whether or not we could produce things locally. So I'm worrying about that. And so I decide that I really want to hurt the United States. So I design a gene drive against insect pollinators of fruit species. And I deploy them just in Florida and California. And let's say they just reduce the pollinator we already have a pollinator problem globally. And now we reduce the pollinator species just for a couple of years by 40%. Imagine what it will do to the food security of the United States. And it'll be very easy to do. I create these insects, put them in a van, come out to some lonely area and release them. That's what pollinate people who have pollinator populations that they sell to people. That's what they do anyway. So I make my vans look like one of these commercial vans. Nobody will question me. First time it doesn't work, I do it again. Just as an ordinary pollinator services company would do, I go and I actually sent out these things that would create gene drives that would make the populations drop. And there's no way I know of that this could be prevented. The only reason it has not, nobody seems to have done it successfully so far, is actually designing these constructs has not been as easy as designing constructs against mosquitoes. But this is a real biosecurity worry that arises from CRISPR-based germline editing that we should be worrying about. And in the case of bees and things like that, luckily, because if you have haplodiploidy, designing gene drives is not that easy. But if you have ordinary diploid species, and those are your major pollinators, then you have some cause to worry. So these are the things that you should really worry about with gene drives, and not worry about designer soldiers or designer babies and all of that. Let me stop here and see where we are. Okay.